Welcome friends, my name is Nayab Ishrat and I am a PhD student in King George Medical University in Biochemistry department. Today I am going to present my presentation on insight into the possible association of COVID-19 with human. So first of all we have to know the origin of this COVID-19. First time on December 30, 3 cases were reported in Wuhan, identified in a small local fish and wild animal market in Wuhan city. The Chinese Center for Disease Control and Prevention reported that a strain of coronavirus that is distantly associated with a severe acute respiratory syndrome or SARS-CoV monophyletically group might be causative agent of the 2019 novel coronavirus. Later on, it was established that novel type of COVID caused the outbreak and was named as SARS-CoV-2 as a result of genetic recombination occurs between members of the same or different coronavirus groups. New viruses emerging from the animal's reservoir and subsequently to human. Moreover, there are 3 new coronaviruses have been emerged as a result of their genetic recombination, which are MERS, that is Middle East Respiratory Syndrome, SARS, that is Severe Acute Respiratory Syndrome, and 2019 and Beta Coronavirus 2019 Novel Coronavirus Acute Respiratory Disease. Here is the structure of the SARS-CoV-2. You can see the structure of this showing Center of Disease Control and Prevention, which was images were given by these. The spikes on the outer edge of the virus particles look like a crown, giving the disease its characteristic name. COVID means crown. So, in depth of the analysis of the genomic characteristics of the SARS-CoV-2, coronavirus belong to the family Coronaviridae that replicate in the cytoplasm of the host cell. It is enveloped positive strand. It is a RNA virus with largest known RNA genomics of 30 to 32 kb. The SARS-CoV-2 genome has similarity to other COVIDs, consisting of minimum 10 open reading frames. The rates of mutation of RNA virus is greater when compared to the DNA viruses, which propounds that COVIDs have a more effective adaptations process for their survival. The ORFs present in 1/3 of the COVID genome codes majorly for 4 different proteins that are the integral part of the viral center. The spike glycoproteins, which binds to the host cell receptor and helps in facilitating the entry of virus into host cell. Envelope protein E, which is responsible for interacting with the membrane protein for formation of the viral envelope. Membrane protein M, which is the primary organizers of the COVID-V assembly to determinant of the viral shape, and nucleoplasmid N, which binds to the RNA genome. Nucleocapsid protein makes a complex with the RNA genome and makes an arrangement of helical capsid, which is found in the viral envelope. The S protein process, the site of viral attachment to the host cell. The small spikes of the hemagglutinin esterase proteins on the membrane are present in some coronavirus virions. The transmembrane proteins M and E are also involved in the assembly of the virus. The virus releases its RNA genome into the host cell after gaining entry into the host cell. It then translates its genome into 2 polyproteins and the structural proteins, which leads to the replication of the viral genome. Here is the, after knowing the genomics of the COVID-19 structure, we can see the life cycles of the SARS-CoV in human lung cell when a human is infected by COVID or SARS-CoV-2, we can see the life cycle of this virus in human being. Coronavirus is most often transmitted by droplets while sneezing and coughing, and its journey begins in the first days after infiltration from the upper respiratory tract. The spike proteins of SARS-CoV-2 binds to the ACE2 receptors. The virion then releases RNA genome into the cell, and translation of the structural and non-structural protein follows. ORF-A and ORF-B are translated to produce PPA1A and PP1AB polyproteins, which are cleaved by the proteases that are encoded by the ORF1 to yield non-structural proteins. This is followed by assembly and budding into the lumen of endoplasmic reticulum Golgi apparatus. Virions are then released from the infected through exocytosis and then ready to infect other humans. The key reservoir and potential interspecies transmission routes of SARS-CoV, MERS-CoV, and SARS-CoV-2 is coronavirus might be infected through the bat, then palm civets, camel through palm civets, SARS-CoV through camel MERS. The probability is one species is pangolin. After the infection, this is transmitted from one to several, and then immediate emergency is required for their treatment and hospitalization. The symptoms given by the WHO in a human being are common symptoms are headache and the body fever temperature raises above 38 degrees centigrade, runny nose, dry cough and sore throat, general feeling of fatigue and exhaustion, severe acute pneumonia, hemoptysis, severe diarrhea, and severe shortness of breath. Here is the total prevalence of COVID-19 cases till April 14, and it can be seen it governs North America 16%, Asia 48%, Europe and 1% in Africa. What are the possible drugs so far for this treatment of the COVID-19? Scientists are doing all effort to get drugs soon in the market. In the meanwhile, the drugs so far come to the knowledge is remdesivir is considered the most promising antiviral agent. It works by inhibiting the activity of RNA-dependent RNA polymerase. A large-scale study is investigating the clinical efficacy of remdesivir. The other excellent anti-influenza is RDRB inhibitor. Favipiravir is also being clinically evaluated for its efficacy in COVID-19 patients. The protease inhibitor lopinavir LPV-RTV alone is not shown to provide better antiviral efficacy than standard care. However, the regimen of LPV-RTV plus ribavirin was also shown to be effective against SARS-CoV in vitro. Another promising alternative is hydroxychloroquine 200 mg thrice daily plus azithromycin 500 mg on one day followed by 250 mg twice daily on day 25, which showed excellent clinical efficacy on Chinese COVID-19 patients and SARS-CoV potency in vitro. Monoclonal and polyclonal antibodies in the treatment of SARS-CoV are under investigation. Avoiding the prescription of non-steroidal anti-inflammatory drugs, angiotensin converting enzyme inhibitors or angiotensin II type 1 receptors block is advised for COVID-19 patient. What are the challenges to the treatment strategies? The major target for COVID drugs and vaccines is the spike glycoprotein, which is the primary inducer of antibody neutralization. Till date, the attempts to develop effective vaccine against human COVID infection, such that MERS and SARS, have not been fruitful, and hence the scientific community has not been able to develop any licensed antiviral treatment or vaccine for these infections. One of the most important factors for antiviral drug development is the demand in the market, which lasts only for the period while the outbreak lasts. The issues with antiviral drug is that when it is ready, there are no patients for clinical trials. Due to these issues, the pharmaceutical companies lack interest. The mouse does not show interaction with MERS or SARS virus due to the differences in the ACE2 receptors. Hence, the mouse needs to be engineered first to delete the mouse ACE2 and acquire the human ACE2 in place of that, so that we can do clinical trial on rats. References. Thank you, and be alert and not alarmed. Thank you for watching my talk.